use this. Advantages and disadvantages. Uh, Hopefully, it will be clear with the passage of time. Uh,
सर ये
what kind of advantages and disadvantages? What do you think? What would be the advantage of the microscope?
लाइट माइक्रोस्कोप
Yes,
सर पोर्टेबल
it's an advantage. Yes, you can take it with you wherever you want to use. It's easy to use. It's easy to handle. So
एंड
these are the advantages that
वी
we
कैन
can say.
एग्जामिन लिविंग स्पेसिमेन
Anything else? Uh, there is a disadvantage in this case that has poor magnified image. The image that, for example, if you have already used your light microscope,
यस
so hopefully you have experienced this fact that once you will increase the resolution or the increase the magnification of the image that you are observing under the light microscope, that image will become blurred with the passage of time. That image will distort. This is the disadvantage what we have observed in the light microscope. Are you getting my point?
यस सर
Okay, what I'm saying, once you will increase the size of the image up to a certain limit, then it would start, distort. So that's why it's a disadvantage of light microscope. In case of, in case of electron microscope, that is very sophisticated microscope, so it's not easy for everybody to use. So that's we can say that's little bit difficult in handling. You need a very specific training on that microscope, then you will be able to use it. Otherwise, any newcomer, anybody who is not much trained on the microscope, specifically scanning electron microscope or the electron microscope, then he or she cannot be able to use it. So you need a specific training. Even it we always have people like who have very much specialization in any one of these microscopes. For example, they can get the PhD degree on it. They can get any other specific diploma on this training. So then they will be able to utilize this facility, and then they will be able to handle it. So another thing, for example, if you want to see something with a shorter wavelength,
यस
in case of light microscope, we have studied that you need a shorter wavelength under which you can observe easily in case of light microscope. Are you getting?
यस सर
If you will increase the wavelength of the light which you are using in the electron in light microscope, then it would be difficult for you to observe the specimen under light microscope. So
यस
these are some advantages and disadvantages that you must know about this, or I don't think so. Nobody will specifically ask you this question, but at least you should know about the difference between these two
यस
types of microscopes.
यस सर
Now we are going directly jump to the cell membrane. Cell membrane is our first topic. That is, we will discuss one topic specifically. That is magnification and resolution. How we can determine this, both of these specific concepts, and then how we can calculate this. But right now we are going towards the cell membrane.
ओके सर
Cell membrane. Right now, I think it's not much clear, but can we do something else like in this way? Oh, yes, fine. skull is not much like, yes, it should be very clear. Now it's okay.
Yes, sir.
Okay, cell membrane. Do you have any concept about it, or
Yes,
is
sir.
what is it?
Sir, it
Cell
is
membrane.
phospholipid bilayer, and
Okay,
it controls the movement of substances in
molecules.
the cell.
What are getting in are the molecules or the substances which are coming out. Is it?
Yeah. Yes, sir.
Cell membrane is the outermost structure or the outermost boundary in case of animal cells,
Yes, sir.
right?
Yes, sir.
And this cell membrane is the second most, the second outermost boundary in case of plant cells. And cell wall is present in this plant cell because that is the outermost layer in case of plant cell.
Yes,
So cell membrane is the, as you said, it's a bilayer,
yes,
lipid bilayer. It's a lipid bilayer, and lipid bilayer you have specified here the phospholipids.
yes, sir.
Commonly, the structure we are using for it, that is phospholipid. There is a specific head, and
And
then there is a specific tail.
tail.
This is a phospholipid, the molecule of phospholipid.
Yes, sir.
Then, then this phospholipid is further combined with each other and to form a layer like this, like this. So, what is the specificity and what is the specific character of this phospholipid? Why this phospholipid is part of the cell membrane?
Sir,
This is
it
the
is
question that.
uh,
Yeah.
the head of the phospholipid layer is hydrophobic, and the tails are, I think, hydrophilic.
Uh, just try to recall it. Are you sure that head is hydrophobic and tail is hydrophilic? Tail is hydrophilic or hydrophobic. You have first we can differentiate these two terms: hydrophilic and hydrophobic.
Water heating
Hydrophilic,
and water loving.
yes, water loving structure. So, what do you think water loving structure should be where? Is it? I mean, it should be clear, very much near to water, and tail that is, if for example that is hydrophobic, so that should be away from the water.
Yes.
In this condition, for example, we can just draw a structure just for our understanding like this. Uh, like this. Just we can get an idea from here. You have tail. These two lines are actually showing you a tail. Definitely, we cannot make it much clear here, but it's a structure that from where we can understand easily. So
Yes.
there is a head. Head is very much near to the water. So remember, there's a difference. Tail is what? Tail should be hydrophobic.
Okay. Okay.
That's why hydrophobic means that is a water heading. If
Yes.
it will be a water heading, as a result, the tail of all these phospholipids will try to hide themselves from the water.
Yes,
Are you
sir.
getting?
Yes, sir.
So that's why the tail is actually aggregated inside of this structure just in order to avoid the water interaction.
Yes, sir.
Are you getting?
Yes, sir.
Okay. Now head is actually, it is very much exposed to the water, right? So it
Yes.
means it is hydrophilic.
Philic.
Got it? So that's the difference. So you can build this concept just by having this in mind that tail is actually getting away. Both of these, the tails from both sides of these phospholipids, they are aggregating with each other, trying to hide themselves, trying to avoid the water interaction. On the other hand, the head is very much exposed to the water. So it
Okay.
means that is water loving. So that's a difference between the phospholipid specific structure that is head and tail. And we can also understand this structure when we will study the next where we will specifically discuss about this phospholipid structure. So right now, it is what we call semi-permeable membrane. Another term that we always use for this that I'm writing here,
Yes, sir.
right, right here. Semi-permeable. I have to choose some other way to uh, write on the board because this board is not very much clear. So that's why I'm not feeling much easy to write on it. So it's a semi-permeable membrane. Another word that we can use, partially permeable membrane. I think both of these terms are very much familiar to you. Is it? I
Yes,
don't think so. It's both of these are different. Semi-permeable. I mean, another word that we can use for it. I'm writing here. Selectively permeable.
yes, sir.
Selectively permeable. These are different terms that we commonly use for uh, we can say the function of the cell membrane. Actually, this cell membrane allows few substances to move in and restrict or bound substances not to get in. This is the difference.
Okay, sir.
For example, uh, you have any charged particle, and that charged particle is trying to get into the cell membrane, into the cytoplasm. Definitely, what is inside of this cell membrane? There is a cytoplasm, and any material that is coming from outside, that material wants to get in,
Yes.
and this cell membrane will decide whether this molecule should get in or not.
Okay, sir.
All right.
Yes, sir.
So in this case, for example, you have any charged molecule, charged substance. So that charged substance will not be able to pass this structure because this phospholipid will not allow it. Because charged structure or charged molecule will be restricted by this cell membrane, right?
Yes,
So
sir.
it's a fact. Uh, Commonly, the fence that we always use as a boundary from any specific area. So now we will talk about its chemical composition.
Okay,
What is
sir.
its chemical composition? It is mainly composed of, yes,
Yes, sir.
it is mainly composed of,
Sir, the phospholipid bilayer?
mainly, uh, what would be the components of this cell membrane?
Sir,
There
phosphate.
will be a protein, okay,
Okay.
and then
So can you repeat it?
what I'm saying,
Yes, sir.
composition of cell membrane. What type of molecules or substances which are present here? The first type is protein. Second is lipid,
It's
and the third type is uh, lipid is there are some proteins here. How these are present, as I have told you, because we have studied the lipid bilayer,
yes,
lipid
bilayer.
bilayer, and how these other two types of macromolecules are present here. So they will be present like they will be embedded in it. We will use the word embedded. So if, for example, you have these two layers,
Yes, sir.
so proteins will be embedded in it like this, like this, like this. Okay.
Okay.
So on the other side, you have a lipid. Lipid, which is present in the cell membrane, that is cholesterol.
Okay, sir.
That is cholesterol.
Cholesterol.
Actually, cholesterol's main function in the cell membrane is to maintain the structure of cell membrane. There is no other any specific function of this lipid. Lipid is actually in the form of cholesterol. Cholesterol is what? It is actually lipid. And this lipid is present other than phospholipid. Phospholipids are definitely, that's the main part of the cell membrane, because that has formed, uh, in this case, that has formed actually two layers, two specific layers. That's why we call it bilayer. So this is what its composition is. And then we can also say carbohydrates. In this case, carbohydrates. Here you can write not protein, here you can write carbohydrates.
सर कार्बोहाइड्रेट्स कहाँ पे प्रेजेंट है इस पे?
Sorry?
कार्बोहाइड्रेट्स कहाँ पे प्रेजेंट है इस पे?
Yes, carbohydrates are present like this. Let me show you. For example, this is a layer. On this layer, there will be a conjugation of this like this. It will be present here. They will form a tail-like structure like this.
Okay.
Like this. So there will be another method by which this carbohydrate will be present. That will be present on this protein as well. That will may also be present on this protein
और सर ये
in this structure like this, right?
कार्बोहाइड्रेट्स का फंक्शन क्या होगा?
Carbohydrate is actually used here. We will study another term, that is what we call glycocalyx. YX, glycocalyx. Its function is, one way its function is to act as a, in this case, act as a receptor.
Okay.
Right? It can also act as a receptor here. And there is a conjugated molecule. We always use a term conjugated molecule. Let me clear it first. Do you know what is conjugated molecule? Conjugated molecule is actually a molecule which is formed by by the combination of two different molecules. आप मेरी बात समझ रहे हैं?
Yes, sir.
So this carbohydrate will perform multiple functions. Like I have told you, it is a structural component. It would help to maintain its structure. It would act as a receptor as well. We will study about this many places there. This carbohydrate will act as a receptor because it is mainly present as a channel. Somewhere we will observe this. The protein channels are also here. So another that we can say, there is a conjugated molecule. You know what is its function? Its function is to strengthen the structure of cell membrane. Are you getting?
Okay, sir.
It's mainly its function here in this case in cell in the specifically in plasma membrane is to provide what? Strength. How? Because it will combine with lipid or it will combine with protein to form a conjugated molecule. We always say that a conjugated molecule is a molecule that is formed because of two different types of molecules, right? So in this case, shortly, there is not much detail in your syllabus, that you don't need enough. You can say detail about this. Another thing that we can say here, its thickness is about 7 nanometer.
Okay, sir.
Thickness of this cell membrane is about 7 nanometer.
सर इस पे एग्जाम क्वेश्चन कैसे आते हैं?
They will give you something like, they will give you a diagram in which they will ask you this question, that differentiate, give the functions of this structure, the name and function of this structure, right? So in this case, for example, they will give you just like here that I have shown you like this. Let me give the name to this structure like here. It is a glycoprotein. Why we call it glycoprotein? because it is made up of two types of molecule. One is protein and the
And
other one is
glucose,
carbohydrate.
carbohydrate.
Carbohydrate is what? We can use this word glyco for it, right?
Yes, sir.
So they will ask you this question that differentiate between these structures and give their function as well. So glycoprotein, I have told you, it is also present here. Like, for example, uh, in this case, for example, here is a protein like this. It's a protein structure, and on which there will be a structure that is glucose. Number of glucose molecules are present here. So in your paper, they will ask you, we will discuss some questions at the end of this topic. So you will understand what type of questions are here. Questions are very simple from here, that they will give you a structure, different types of structures, and they will ask you differentiate these structures. You have to name them and as well as give some function as well, right?
Okay, sir.
So I have told you the composition as well. So that's all I think about this structure. We don't need enough detail about this topic. So another thing you must know about the functions, and in this case, cell membrane, uh, we have talked about that it is made up of two layers of lipids in which proteins and uh, specifically carbohydrates, they are embedded, right? This carbohydrate may be directly embedded or it may also be present on the protein. This is one thing. And the second point that you need to understand, we call it selectively or partially permeable membrane, right?
Yes, sir.
Why we call it? Because we can, infer that because some specific molecules are allowed to get in, or some specific molecules can easily pass to this cell membrane, and many other molecules which are large in size, which are charged cell types of molecules, they will not be able to get in. They will be restricted by the cell membrane.
Okay, sir.
And then what is glycoprotein? Another term we can use, glycolipids. One term we can uh, learn here more, that is glycolipids. Can
It's,
you differentiate glycoproteins and glycolipids?
sir, glycolipids में fat molecule भी होगा.
There will be lipids, and those lipids will be conjugated with,
Carbohydrates.
they will be conjugated with,
Carbohydrates.
yes, they will be conjugated with carbohydrates. And what are glycoproteins?
Sir, proteins conjugated with
It is the
carbohydrates.
conjugated molecule of two components. One is protein and other is
Carbohydrate.
carbohydrate, right? So this is all about the cell membrane. So another structure that we are going to study before going to talk about the internal structure of this cell, that is cilia and flagella. Okay. Cilia and
Flagella.
flagella. Okay. Cilia. Uh, how we can differentiate these two structures? At first, what is cilia, what is flagella, and how they functions in plant cells? That's shortly we should know about it. What it is, both of these structures are what in case of cell? Extension of cell. I will
Okay.
use the word here, extension. Extension of
Cells.
cell surface.
Cell surface.
Extension of cell surface, right?
Yes, sir.
Where? In case of both unicellular and
Multicellular.
multicellular. Remember, both of these structures, cilia and flagella, these are just the extension. What do you mean by this extension? Extension means any elongated structure like this,
Okay, sir.
or just the protrusions of the very small structures like this. Right?
Okay, sir.
Okay. So, as we have observed in case of just like you can say a wiper which is present just outside of the vehicle, like we commonly use on the glass, and that the way that wiper is moving on the glass on the front side of the vehicle, in a very similar way, this cilia and flagella both are working. Like how they will move or how they will be present here, there we can say they can move like a viper. They can be a like a let me write here. Hmm, there is not enough space here. Running time out of time. Why? How much time do we have in this meeting?
Sir, 9 minutes.
Nine, nine
Nine.
to 10 minutes, sir.
Yes, sir.
Okay. Right. So, for example, there is a bacterial cell here, like this. It's a bacterial cell, and there are some protrusions or the viper-like structures which are present outside. This is how uh, specifically the flagella are present, right?
Yes, sir.
With lip. So why we call it? Because from this base they are always attached, and they cannot move completely from one place to another place. They can just like a viper. Its initial point at the root is always connected with something specific, that is fixed. We can say its base is always fixed,
Okay, sir.
and the other structure which is elongated outside that can only move.
Okay,
And yeah, let's talk about flagella first. Long, they are long and few in numbers.
okay, sir.
Few in
Numbers.
numbers. I mean, there will be 2, 3, 4, not more than this,
Okay, sir.
right? Somewhere the bunch of 5 or 6. Okay.
Okay, sir.
While cilia is shorter
Mm-hmm.
and more in number.
Okay, sir.
Cilia is, I'm
Short.
writing here, shorter. How? Like this. These are the structures, like this. Very small structures, not like the, although both have the same sort of composition, but their size is very much different. A size means one is elongated, the other one is shorter. So cilia is shorter and more in number.
Okay, sir.
More in
Number.
number. For example, I'll show you a diagram on which the cilia are present. Like you have a structure here that is again any specific structure on which like this. More in number. That's why they are sometimes they are present on the whole body, entire body from outside of any organism, right? There are many like paramecium, we will talk about this next somewhere. But here, this is a structure of cilia. Here is cilia. These are cilia. And remember, one is cilium. Singular is cilium actually. We are using here plural word, so that is cilia. Number of cilia means there are many ciliums which are present here.
Okay, sir.
Another word that is, that is flagellum. Flagellum is for one. So we are using flagella just because there are a number of 2 to 3, 4 number flagella are present here. Right, Ji?
Okay, sir.
Okay. What is its composition? Composition. Have you talked about somewhere microtubules? You know
Mm,
about these microtubules, what they are?
yes, sir.
What type of structures can you have in your mind, that what type of structure specifically these microtubules, what they are
Uh,
like tubules, as we are using the word tubule, it means it's like a tube.
yes, sir. There are different tubes connected together, I think.
Yes, like here, like this, like this,
Yes, sir.
like this, right? So
Yes,
it's a microtubule. Are you getting? Number of tubules which are connected with each other. Mostly they are triplet in form. Uh, so the composition of both these structures is microtubules, and these microtubules are attached with from the base with cell. Yes, cell
membrane.
membrane. Definitely, the base. It is, for example, this side is base. So this side is attached with the cell membrane, right जी? So उसके बाद हम इसको क्या कह सकते हैं? So numerous small hairs like structures, flagella, very few. I have to already discussed this specific concept. So hmm, it's very unique, although we don't need enough detail, but it is composed of about 600 types of proteins. You just imagine very different types of proteins which are present in these structures. So it means a very versatile sort of structures.
Okay, sir.
Can we locate in case of humans where these cilia are present?
G?
Cilia in human beings,
Uh, s-
trachea,
yeah. Okay.
trachea in your respiratory tract, gut, digestive system.
Yes, sir.
These, I'm talking about cilia, not flagella, right? Flagella is present in many unicellular organisms.
Okay.
Uh, but in case of human beings, we are talking about, so this cilia is actually present in the gut and as well as in the trachea of living organisms, right?
Okay, sir.
Uh, where this flagella would be present? It is present in motile bacteria, remember.
Motile bacteria. Okay, sir.
Why? What is its function? Its function is actually motility. This flagella or these types of flagella which are present in bacteria, they actually help to move the bacteria or any organism on which it is present, right? So most of the time, these flagella are present in bacteria.
Yes.
Cilia is absent in bacteria, remember. Cilia are not present in bacteria. They may ask you in your MCQ. So you must keep in mind, cilia
Okay.
are absent in bacteria. And what is the structure which is present in case of bacteria? That is flagella,
Flagella.
right?
Yes, sir.
And cilia is even also not present in other prokaryotes. So do you have an idea of what is the difference between prokaryotes and eukaryotes?
सर प्रोकैरियोट्स 70s राइबोसोम्स होते हैं और ये
Yes, we can simply we can say membrane bounded organelles are present
and
in case of
non
eukaryotes, and
non
membrane less are the organelles without membrane present in the cell or unicellular specifically. So they are called prokaryotes, right? Example, bacteria. Bacteria is prokaryote or eukaryote?
sir, it's
Prokaryote, na?
prokaryote.
Because uh, bacterial cell is prokaryote because in the cell of bacteria there will be no membrane bound organelles.
Yes, sir.
Nucleus is not bound. Nucleus is not present in the nuclear envelope in case of prokaryotes, but
Yes,
nuclear envelope is present in case of eukaryotes.
yes, sir.
Are you getting this point?
Yes, sir.
Okay. So I have told you how cilia are present. For example, we have uh, these two types of cells. So like this, these are cilia, like this, right? So it's a bacterial cell. So these are the flagella like this. Both the function of both these types of structures is what? Movement.